lot for the very nice introduction. So it's really my great pleasure here in the Dalhousie lecture series to introduce to you maybe an environment, a marine environment that you are not so familiar with. And maybe a few words initially on where I come from. So as Boris said, so I'm from GEOMAR Kiel. So what is GEOMAR Kiel? So that's, if you will, the German Ocean Institute. So we have grown rather large in the past decade. So we at the moment are about 1,000 students, staff, and faculty, and our budget, including third-party funding, is about 100 million US dollar. I hope you don't feel offended that I have the US dollar here, not the Canadian one. And so my research unit is marine evolutionary ecology, and our motto, if you will, which I borrowed from the late Winfried Lampert at the Max Planck and Plön, who borrowed it again from Charles Krebs, is that natural selection is ecology in action, that shows this intimate interweaving of ecological and evolutionary processes. And for those that are not so immersed in evolutionary ideas, I would like to make that clear from the onset that evolutionary adaptation in the strict sense has nothing to do with acclimation, but it is rather a population level process. So tanning, if you go into the sun and your skin gets a bit darker, that's a form of reversible phenotypic plasticity that could also be under the umbrella of acclimation. And then we also see forms of irreversible plasticity during the ontogeny of organisms. For example, those fascinating spines of water fleas that they only form when they grow towards adults, when the pond has fishes. It's a response to fish cues not to be eaten that quickly, whereas they don't if there are no fishes in the pond. That again shows the difference between phenotypic plasticity at the individual level versus population level process, which is evolution. And the first one who really conceived that population level idea, that there is a strong difference in the reproductive success ultimately of members of the same animal or plant population, that was Charles Darwin, of course. And we say that if there is a stronger reproductive success and the carriers of some traits, that these individuals are then positively selected, which means in turn that at the population level, the mean fitness increases with time. And if we look at the genomic or epigenomic underpinnings, that means that allele frequencies change. That was evolution 101. In 2 minutes, let's now focus our view on the marine or the coastal ecosystem. So where is it that really the humans fear the Anthropocene influence, if you will, and the ocean meet? That's of course at the coastlines. Many of you will know that a large proportion of the human population is living very close to the coastline, which means that many of the human pressures, they are particularly strong along the coast. And what does that mean then in turn for ocean basins that are surrounded by coastline and by very intensely populated areas, such as the Baltic Sea in Northern Europe? That means that those oceans, we would predict that those oceans are particularly exposed to a diverse set of human pressures, and that's exactly the case with the Baltic Sea. In addition, it has a relatively shallow medium depth. That means that the water volume is relatively low. On the other hand, its openings, and I hope you can see my cursor now, its openings to the open North Atlantic are relatively shallow. So we predict that the water exchange time with the Atlantic is about 80 years of an average water parcel in that sea. At the same time, the Baltic is very intensely researched. So there are very good long-term data series, and there are also European Union-driven research programs. For example, there was the BONUS program, a science program for the better future of the Baltic Sea region. And within this program, I also ran a larger scale project, and together with 6 other projects, so that, if you will, was a project of projects or a synthesis of already synthetic projects, we came up with the idea, and during these writing workshops, that we can feature, or that we should much more prominently put forward the Baltic Sea as a time machine for the coastal ocean. So the Baltic Sea in a way is not an oddity, but it can serve as a very good bet model, if you will, for maybe the future of some of the coastal seas we are predicting for the decades to come. So why is that? I run you through some of the environmental changes and also at the same time selection pressures that we expect for the next decades to come. For example, here, the warming rate of the Baltic Sea has been particularly fast, in particular compared to the open North Atlantic. So it's about 3 times the global average. Of course, there are other areas like the Black Sea or here the Eastern Mediterranean that can keep up. But as you will see soon, the Baltic is special in that in kind of a decathlon in many of the abiotic pressures, it stands out as being ranked among the top contenders in a very bad way. A second abiotic feature is ocean acidification. So here you see the PCO2 in microatmospheres, that's about ppm in the atmosphere across a summer season of the water actually close by next door to where I'm sitting right now in GEOMAR Kiel, southwestern Baltic Sea. And we do see that we can, that we attain microatmosphere values above 2000 in summer in the Kiel Bight, whereas this is the window predicted for the global ocean in the year 2100. So we are easily exceeding this right now. A bit more formal analysis here. So here the Baltic Sea, in particular here where we are, the southwestern Baltic, and its range of values is compared against other permanent stations, mainly in North America actually. And again, we see here that both the mean but also the variation stand out where we are. So we have in a way a naturally PCO2 enriched laboratory where values are already now much, much higher than they are anticipated for the ocean 2100. Same applies to oxygen. So the Baltic Sea is one of the worst examples for increasing deoxygenation. And again, thanks to these long-term time series, we can get a handle on that. So if we only went back 20 or 30 years, we would completely miss that signal because the year-to-year variation is enormous owing to the very strongly varying influx from the North Sea. So what you see here is in black, those are the small pitches that were in Oxic around the turn of the previous century, so 1900 to 1910. Now the decade 2000 to 2010 is the reddish area. So we see that the anoxic areas have increased tenfold or more than tenfold in the past 100 years. And finally, in terms of anthropogenic pressure, of course, we also have fisheries. Here, one of the most striking examples of declining catches, and you will know that very well in Canada, that you can really destroy a cod stock easily and it will rarely come back. That's the example of, again, the cod stock next door of the GEOMAR Kiel. So here we see a continual decrease of the landings. I could also show you the fisheries-induced mortality, or the recruitment looks all really abysmal. And I would also like to point out to these bars here in the end. So what you see here in green, that's the estimated recreational catch. So at the moment we have a situation that the stock is so low that the recreational fishery catches between 1/4 and 1/3 of the total catch, which is clearly not a sustainable situation. Summing up in that synthesis paper that I can of course recommend because it really summarizes the knowledge on the Baltic Sea. To sum up, if we look at other ocean areas that are remotely comparable, which you see here on the left-hand side. So we compared the Baltic Sea situation to the North Sea, Mediterranean Sea, Black Sea, Gulf of Mexico, East China Sea, and Barents Sea. What stands out here in terms of the Baltic Sea is that you see a lot of red, so really bad environmental status, or at least a yellow. So collectively, again, it's a multi-pressure, it's a very bad multi-pressure situation. However, the second half of the synthesis we've made is that counteracting those multiple pressures, we also have a quite nicely working regime in terms of monitoring, research, and governance activities. So if we really look at the research activities, and I just presented to you some synthesis results from that bonus effort, but also at the monitoring activities, we have actually a very good situation in the Baltic. So why is monitoring important? Of course, it is important, first of all, to figure out that there is a problem in your marine environment. But also if you do an intervention, you would like to see if there's some success. And for both of that, you need long-term data series. And also the governance structure, I just would like to mention briefly verbally. So we have 9 different countries neighboring the Baltic, including 2 political blocs. So we have Russia, and then we have the European Union. But of course, not all states have been in the European Union before. For example, the Baltic states and Poland only joined in the 2000s. But so a priori, you would predict, so this is a really difficult situation to manage, right? To get 9 different countries with their environmental policies integrated. But in fact, the Baltic did that in a very good way. So there's HELCOM, the Helsinki Commission. I will not go into any further here. I just would like to show you 3 of the management successes that happened owing to pretty strong management interventions. So first of all, we witnessed the return of at least some of the bird and marine mammal top predators. Some of the white salmon populations returned. And we also saw a peak of the nutrient pollution in the 1980s to 1990s. And since then, at least, there is some curbing of the nutrient input, which, however, and that's really of concern, has stalled in the last years. And that's probably a result of increasing warming that enhances remineralization and the runoff of nutrients from agricultural lands. So now I hope I have briefly shown you the very particular selection environment of the Baltic Sea. Now let me introduce to you the Baltic as the evolutionary time machine, which is actually the title of my lecture today. So if you look at the Baltic, it's for all marine organisms a dead ending. And it's also, compared to the open North Atlantic, for example, a relatively small marine area. So that means that already in a synthesis that Kerstin Johannesson and Carl André published in 2006 in Molecular Ecology, if you compare of all the species where you have sister populations in the North Sea, North Atlantic versus the Baltic Sea, you do see that this pairwise comparison of genetic diversity at molecular markers is almost always tilting towards the Atlantic population that shows a higher diversity, which is predicted. So we know that we have less genetic diversity for adaptation to work out in populations of the Baltic Sea. In some cases, we even have the extreme situation that that macrophytes, which are the foundation of many ecosystems, such as these Fucus bladderwrack species, they're entirely clonal. So they don't do sex anymore, and they proliferate by vegetative growth. Fucus radicans has also been described as a new own species that occurred only by asexual reproduction. I'm not going into the discussion now if that counts as a real species, be as it may. So in large proportions of the central and northern Baltic, we have the bladderwrack reproducing only asexually. Another really interesting example are these mega clones in the eelgrass that also at first sight seem to be genetically invariant. However, some recent work by our group showed that somatic mutations do seem to play quite a significant role in generating new genetic variation that occurs via somatic mutation and distributing them among the branches, among the modules of a seagrass clone. So we're really excited about this possibility because it may pose one solution to the riddle why in those stressful environments genetic diversity doesn't seem to be important at all. So that flies in the face of many of the conservation genetics dogmas. And one of our propositions is actually that they may not be as genetically uniform as people previously thought. So nicely, we could team up and get some money by Human Frontiers of Science program to explore that in more depth, also together with coral researchers and cancer researchers studying similar processes in these systems. But let me now turn over, let's say, to normal populations that reproduce sexually. And one change of environment or strong gradient, I should say, in the Baltic, I actually didn't yet go into detail. So the Baltic Sea is one of the largest brackish water areas in the world ocean. So it can serve as a very nice natural laboratory again to do studies on other predicted quick, rapid temperature changes in salinity that we are expecting in the global ocean. So in particular in the tropical and subtropical areas, an increase of salinity is expected, whereas in many near polar regions or temperate regions, owing to enhanced freshwater runoff, melting glaciers, and so on, a decrease of salinity is expected. And so now enter the Baltic Sea again as a model, and enter the three-spine stickleback as a nice marine to freshwater model fish. We have this very pronounced salinity gradient in the Baltic from almost freshwater in the Bothnian Bay to a very steep transition zone where we are. So this is here would be Kiel approximately, to full marine outside the Baltic Sea. And so we can use that spatial gradient as a very nice space for time proxy to study rapid adaptation to, in this case, decreasing salinity. We also know from previous work, previous experimental work, that there is local adaptation. So in particular, low adapted stickleback populations from inside the Baltic Sea, they don't perform very well under high salinity. So we do see some home site advantage here for the stickleback. However, full genome sequencing that has been conducted a couple of years ago showed an extremely low genetic differentiation despite those quite pronounced phenotypic differences. And now I would like to introduce the extended eco-evolutionary synthesis to you. So what does this now mean? This means that there are possibly other hereditary processes that add to the standard population genetic model. So if you will, that this is the normal cycle of population level adaptation that Darwin saw for the first time, or conceived for the first time, although he had absolutely no clue where the hereditary information was residing. The DNA, of course, he had no clue about that. So we now think that there's an additional level of variation that's commonly called epigenetic variation. Some of you may already have come across that. If you're evolutionary biologists, some others through the public media. So we also now know that this affects humans. For example, famine and really strong stress events, they can run through several generations and still impact the grandchildren of the affected parents. So the key question here is if these additional layers of heredity, if they are working just like normal DNA-based mutations, and there's evidence that they do, but there's also a class of epigenetic changes that are precisely introduced by altered condition. And we are not looking here at famine, evidently, but in the stickleback case, at salinity change. To show this in a little bit more detail, and actually postdoc Britta Meyer and ex-PhD student Melanie Heckwolf, they were doing that work. So we are looking at this addition of a methyl group to a cytosine, and that often drastically changes the gene expression of affected genes or promoter regions. And to bring this into the context of plasticity that I introduced in the beginning. So we think that there is either a plastic response that manifests itself in a methylation change. So that would then be inducible methylation, or a stable one where DNA methylation marks are just accumulating or dissipating in a random fashion, just like normal DNA mutations. So far, so good. So until we did the study for wild population, that was more or less a theory, but there were no field data. So we used the stickleback example and took 3 populations from divergent salinity, so almost fully marine, 33 PSU, mid salinity, so where keelless, like 20 PSU, and 6 PSU, so very close to freshwater. And in a study that both involved measuring the methylation marks in the wild, so how are they distributed across salinities, and performing a 2-generation acclimation experiment that here in this case only shows the treatment going from 20 PSU to 6 PSU. Melanie and co-workers were able to show that yes, there are some DNA methylation marks you will not be able to change compared to the wild situation, but others are being induced over these 2 generations to resemble the existing gradient in methylation that is responding possibly to salinity much more closely. So in other words, inducible methylation most probably contributes to salinity adaptation via transgenerational plasticity. And that also makes sense if we look at where those methylation marks are. They are enriched in areas that are responsible for osmoregulation. So a very nice story that I can recommend to read in more detail. Let's now move from salinity to another major pressure, that's ocean acidification. So, and let's here look at the cod that we already saw how abysmal the Western cod population is. Maybe only focus at the moment at the temperature, salinity, oxygen profile, and pH profile in the central Baltic Sea, where the Eastern cod population is residing. What you see here, where the cod is spawning in 60 to 70 meters depth, we have a pH of only slightly above 7.2, 7.3 typically. So that corresponds to a PCO2 that is 1,800 to 2,300 microatmospheres. Yeah, to bring that into perspective, so under the worst case scenario of the IPCC report, we would attain approximately 900 microatmospheres in the saturated surface ocean. So we have here more than double. And again, this would be a typical pH profile of the open North Atlantic. So here only the top 300 meters, right? So we see there's a slight drop because we have a slight oxygen minimum zone here in the deeper water. But by and large, the pH values stay at 7.8 to 7.9 in the North Atlantic. So now knowing that cod exists, of course, in the North Atlantic and in the Baltic Sea, we can derive some specific predictions here. We would say, since cod is existing in the Baltic Sea, those larvae should be pretty much tolerant to those high PCO2 values, and not surprisingly, they are. So that's from the PhD work of Andrea Frommel. So initially, of course, there was a very disappointing result. Of course, PhD students, they only would like to see strong effects, but in hindsight, of course, it's a very cool result because it essentially shows, regardless which PCO2 you apply in the experimental setup, there's no significant effect in terms of hatching rate of the eggs or survival rate. However, cod from the areas that normally do not experience these really low pH values, AKA high PCO2 values, they are very well sensitive. So these are the data from Öresund cod, so that's at the exit or entrance of the Baltic Sea, and of Barents Sea cod, so the Scray actually. And both of them were shown in very skillful experiments done by PhD student Martina Stiasny under the lead of Katriona. They showed in twice completely independent experiments that we see a drastically increased mortality, so a doubling basically of the mortality of juvenile cod, which is of course of concern. And if that materializes in the wild, it would have pretty dramatic consequences for recruitment. So to sum up this story here, so we have a local population of cod in the Baltic Sea that needs to spawn in areas that have a really abysmal ocean acidification situation, which is, by the way, driven by the bad oxygen condition. And the oxygen is converted in CO2. I come to that a little later. But nevertheless, they're able to reproduce successfully. However, we see indirectly from comparison with two other stocks that are very well affected by those values that some pretty strong local adaptation must have taken place. The next example in that arena comes from the group of Frank Melzner. So, and it deals with a calcifying organism, the blue mussel, which you all know. So the blue mussels, of course, have a massive problem because they spawn exactly at the time where very often the Kiel Fjord has again a very high PCO2 that's parallel to the levels we saw in the deeper waters of the central Baltic where the cod spawns. And again, they wanted to figure out how can we compare, or what, so the prediction again would be, of course, if you take now mussels from a more benign PCO2 environment like the North Sea and compare them to those from the Kiel Fjord, we would expect again that there's some local adaptation with respect to the tolerance of the settling larvae to PCO2. Again, in a way that's a space for time approach. And exactly this is what happens. So if you take Baltic Sea mussels, the survival of the newly settled spat of the mussels is indistinguishable between the controlled PCO2 and the really high ocean acidification levels, whereas in the North Sea, if you take North Sea mussel, there's a dramatic decline of the survival. And then they also wanted to find out if there's any variance between the mussel families in terms of their resistance to, or in terms of their tolerance towards high PCO2 values, and indeed they found some. So that's again a cautiously positive message here. So there seems to be additive genetic variants that would allow further adaptive evolution to withstand really bad ocean acidification levels in the blue mussels. So now, having spoken about this physical pressure, the ocean acidification, let's turn to fisheries. So in fisheries, it's now accepted since about 10 years that fishing is not only demographically a problem if you fish too much, but also evolutionary. It will elicit very strong evolutionary responses, given that many life history traits such as length or age at maturity, or also growth rates, have a quite high heritability. So yeah, so this is, and that's actually science historically very interesting, because fisheries ecologists were fighting that idea teeth and claws since about 2000, 2005, and then basically everybody agrees. Still lacking is finding the smoking gun. So we still do not have really direct evidence that something on the genome or at the population level in terms of gene frequency has changed in response to fishing. And this is something we would like to set out again with the Central Baltic cod. So why is that population so very nice to study exactly that? It's because it's in that dead end and it's hardly exchanging any gene pool with the populations outside the Baltic Sea. So we know from marker studies that this is a really confined population. It's not exchanging, so it's really sitting in the Central Baltic Sea now for decades, but it's changing a lot. So this plot here shows, in particular, look at the red line here. It shows the length at maturity, how it decreased both in males and females about equally strong in the last 25 years. So that's a very dramatic decline from about 40 centimeters to about 20 centimeters of length. So now, of course, all the fisheries people here will say, yeah, but you don't know the age of maturity. Absolutely right. Due to the odd salinity environment of the central Baltic, we cannot age them at the moment, but we will be able to age them very soon through some colleagues at DTU Aqua. So we will know at one point what the age at maturity looks like. And yeah, so we have a running project where we will do full genome resequencing of a whole time series to find out if we can find here the smoking gun. So a change at the population level of the allele frequencies in these cod that have changed so dramatically in terms of their mean phenotype. Yeah, no evolutionary process would be complete without speciation. And I already mentioned this asexual speciation mode, this Fucus radicans, that is an asexual offspring of Fucus vesiculosus that you also have in Canada. So it really spreads over hundreds of kilometers as an asexual offspring, never producing sporangia or anything else. And of course, it's debated if this is then a species or not. But as I said, be as it may. So we have these asexual propagating new forms that also phenotypically look very different. Another speciation that probably every ecologist would buy is a true speciation is in flounder. So I had already mentioned that the deep sea oxygen situation in the Baltic is bad. It's historically bad, but it also became much worse. And therefore it was a big surprise when Paolo Momigliano in the Juha Merile group came up with this study here, showing that the flounder that's normally a benthic spawner has developed a second good species, I think, that has pelagic eggs in the central and northern Baltic Sea that's reproductively completely isolated from the ancestral flounder species. And they think that the speciation really took only in the order of 100 years or less. And of course, since the reproductive mode is so very different, the gene pools are completely separated now. So under the Ernst Mayr definition of the biological species concept, that would perfectly fall under that definition. Okay, to sum up, so the Baltic is, in terms of the abiotic drivers, one of the one of the area of the coastal areas that are most impacted. So either driven by human activity such as warming or PCO2 dissolution into the waters, and or nutrient pollution that then ultimately reduces oxygen in the bottom layers owing to enhanced respiration by microbes. But there are also natural gradients such as salinity that we can use to do experiments in a space for time fashion with respect to expected salinity changes in the rest of the coastal world ocean. To wrap that up more verbally, so we have this high pressure complex management framework, but still the Baltic Sea neighboring states, they were able to come up with a political framework that allowed some management that was effective. Helping there were, of course, long-term time series that were often started about 100 years ago that show both the deterioration of the ecosystem, but also intervention successes. So that was the basis for a science-based management. And these management successes, including the governance regime, they can serve as a role model for other world regions where you have multiple states neighboring it that may also belong to different political, let's say, orientations or blocks. The elephant in the room, however, and I didn't go into much detail here, but that's more and more apparent, both for the nutrient problem, but also for the fisheries management, is climate change that is increasingly jeopardizing the successes of regional management. In terms of the Baltic as an evolutionary time machine, we witness, if you will, a Baltic paradox. So even although populations are usually less diverse than their North Atlantic sisters, we nevertheless have many examples of quite strong local adaptation that possibly has also worked out in a quite swift rate. Although we often don't know really the rate, we can just give an upper limit, which is 5,000 years, the existing age of that brackish water body. The Baltic, in many respects, is ideal for a space for time experimental design to study evolutionary adaptation. I've shown you the first case of epigenetics, really mediating a grading of salinity adaptation that looks as if both interact, the transgenerational epigenetic marks and those that are existing already out in the wild that are not easily changeable through experimentation, and also clones that we're often witnessing in the Baltic in terms of macrophytes, fucus or seagrass. They may actually be not as genetically dead-endish as people always think, but they may be able to evolve through somatic mutations. And with this, I'm at my end. I will be ending the presentation